Fellow learners, greetings of the day. Welcome to SWAYM course on microbial technology, week number 4, lecture number 14. Myself, Devashish Sahu, currently working as research head, BioNovel Life Science Private Limited, Bangalore, having more than 13 years of research experience in biological field. Today, we shall learn about an important aspect of bacterial growth phase, its importance and its mathematical implications. But before we start our current session, let us have some throwback of previous lecture session and possible learning outcomes, that is kinetics of the growth. Kinetics of the growth, microbial population exhibit dynamic growth pattern that are fundamental to understanding their ecology, industrial application and medical application. Central to this understanding are concepts such as generation type, doubling time and mathematical expression of exponential growth. Microbial population dynamics study is an important aspect of this particular session. Microbial population consists of vast number of individual cells that interact with their environment and each other. Understanding the dynamics of population growth is essential for predicting population size, optimizing industrial process and managing microbial communities in natural and engineered system. So, coming to the current session, that is based upon the mathematical expression of exponential growth phase. The subject that are discussed in this current session focuses on introduction to log phase, its importance, physiological and metabolic processes, factor influencing the log phase and its implication. And we shall discuss more detail on mathematical application of exponential growth phase. Introduction to growth of microorganisms. So, let us know what is actually the growth. Growth in microorganisms refers to the increase in number not in the size. Microorganisms such as bacteria, archaea, fungus and some protista, they reproduce through various methods leading to population expansion. Now, let us learn about the growth phase. The growth of the bacteria in a closed system such as batch culture typically follow a characteristic pattern consisting of 4 distinctive phases: lag phase, log phase, stationary phase and decline phase, or called as death phase. So, each phase is defined by specific physiological and metabolic changes. Now, let us dive into more detailed aspect of exponential or log phase. The log phase, also known as exponential phase, represent a period of rapid and balanced bacterial growth characterized by exponential increase in cell number. This phase follows the lag phase, during which the bacteria adapt to their environment, to their new environment and prepare for the active growth. Log phase. Understanding this mechanism that drives the log phase is crucial for various fields including microbiology, biotechnology and medicines. This detailed exploration will cover the underlying physiological and metabolic process, factors influencing growth phase and implication of the log phase. So, first point of physiological and metabolic process is DNA replication and cell division. One of the hallmark feature of the log phase is the exponential increase in cell number. This rapid growth is supported by robust DNA replication and cell division process. Bacteria replicate their circular chromosomes using highly coordinated enzymatic machinery. The initiation of DNA replication is tightly regulated to ensure accurate copying of genetic material. DNA polymerase enzyme synthesize new DNA strands aided by other proteins that unwind and stabilize the DNA during the replication. As DNA replication proceeds, bacterial cells prepare for division. The cell cycle in bacteria typically involves several stages: DNA replication, partitioning of replicated chromosomes, formation of division septum and cell separation. During the log phase, these processes occur rapidly and efficiently to maintain the exponential growth rate. The second most important physiological and metabolic process that occurs during this exponential phase is protein synthesis and ribosome biogenesis. The rapid increase in cell number during the log phase necessitates a corresponding increase in the protein synthesis. Bacteria synthesize a vast array of proteins involved in cellular functions such as metabolism, transport, cell wall synthesis and regulation. Ribosomes, the cellular machinery are responsible for protein synthesis, are produced in large quantity to accommodate the higher demand for production of new protein during this process. Then the process of ribosome biogenesis involve the coordinated synthesis and assembly of ribosomal RNA and ribosomal proteins. These components come together to form functional ribosome capable of translating the messenger RNA mRNA into proteins. The rate of protein synthesis is closely linked to the growth rate of the bacteria, with active growing cells synthesizing proteins at a rapid pace to support cellular function and division. The third important physiological and metabolic process is metabolic pathway and nutrient utilization. Metabolism during the log phase is geared towards rapid growth and proliferation. Bacteria adjust their metabolic pathway to efficiently utilize available nutrients, ensuring that energy and building blocks are available for biomass synthesis. The metabolic response varies depending upon carbon and energy source available in growth media. For example, in presence of glucose, bacteria typically utilizes glycolysis to break down glucose into pyruvate, generating ATP and other precursor molecule for biosynthesis. The TCA cycle, alternatively called as tricarboxylic acid cycle, then oxidizes pyruvate to produce additional ATPs and reducing equivalents NADH and FADH2, which are crucial for energy generation and biosynthetic processes. In environment where alternative carbon sources are present, bacteria may induce specific catabolic pathway to metabolize these substrates. For instance, the induction of lactose operon in E. coli allows for the utilization of lactose as the carbon source through the production of beta galactosidase and other enzymes involved in lactose metabolism. Then another important physiological metabolic process during this exponential phase is cell wall and membrane synthesis. As bacteria grow and divide during the log phase, they must synthesize new cell walls and membrane components to maintain the cell integrity and accommodate cell division. Peptidoglycan, a unique polymer found in bacterial cell wall, is continuously synthesized and remodeled to accommodate cell elongation and division process. The process of peptidoglycan synthesis involves the coordinated action of enzymes responsible for glycan strand elongation and cross linking. Penicillin binding proteins called as PBPs play a critical role in peptidoglycan synthesis, as they catalyze the final stages of cross linking to form a stable cell wall structure. Similarly, bacterial membrane undergo dynamic changes during the exponential phase. Lipid synthesis and membrane protein insertion are essential for maintaining membrane integrity and function. Lipid bilayers are synthesized using precursor molecule derived from metabolic pathway, while integral membrane proteins are synthesized and inserted into the membrane to facilitate transport, signal transduction and other cellular process. So, now we will discuss what are the factors that influences the growth rate during the log phase. First is nutrient availability. The availability of nutrient in the growth media directly influence the growth rate during the log phase. Bacteria require essential nutrients such as carbon, nitrogen, phosphorus, sulfur and various micronutrients to support growth and metabolism. Limiting concentration of this nutrient can restrict the bacterial growth, leading to reduced growth rate or prolonged lag phase. Optimal growth conditions typically include a balanced supply of nutrients that support maximum growth rate. In industrial and research setting, Nutrient rich media are often used to promote rapid bacterial growth during the log phase and maximize biomass production. Second part that affects the growth rate during the exponential phase is the environmental condition. Physical and chemical factors such as temperature, pH, osmotic pressure and oxygen availability profoundly impact bacterial growth during the log phase. Bacteria exhibit optimal growth within the specific range of these environmental parameters, with deviation from optimal condition leading to reduced growth rate or growth inhibition. Temperature in particular has a significant effect on the bacterial growth. Most bacteria has optimal growth temperature range, beyond which the enzymatic activity and cellular functions may be impaired. Psychrophilic bacteria thrive at low temperatures. Mesophilic bacteria grow optimally at moderate temperature. Thermophilic bacteria prefer high temperature. pH level also influence bacteria conditions, growth conditions, as changes in pH can affect enzyme activity and membrane integrity. Oxygen availability also plays a critical role in aerobic and anaerobic growth. Aerobic bacteria require oxygen for cellular respiration and energy production, while anaerobic bacteria utilizes alternative electron acceptors such as nitrate or sulfates. The availability of oxygen in the growth medium determine types of metabolic pathway bacteria can employ and influence their growth rate during the log phase. Third one that influences the growth rate during the exponential phase is the quorum sensing and cell density. In some bacterial species, growth rate during the log phase is influenced by cell to cell communication mechanism, alternatively called as quorum sensing. Quorum sensing involves the production and detection of signaling molecule, these are the autoinducers, by the bacterial cells. As bacterial population density increases during the log phase, the concentration of autoinducers also rises. Upon reaching a threshold concentration of autoinducers, bacteria can initiate coordinated behavior such as biofilm formation, virulence factor expression and change in metabolic pathway. Quorum sensing allows bacteria to synchronize their growth and behavior in response to population density, optimizing resource utilization and competitive fitness during the exponential phase. Conclusive remark on log or stationary phase. The log phase of bacterial growth is characterized by exponential increase in cell number driven by robust DNA replication, protein synthesis, metabolic activity and cell division. Bacteria rapidly adapt to their environment, optimize nutrient utilization and adjust metabolic pathway to support growth and proliferation. Factors such as nutrient availability, environmental condition and cell density influence the growth rate and physiological response during the log phase. Understanding the mechanism and implication of log phase growth is essential for various fields such as microbiology, biotechnology, medicines and environmental science. By elucidating the complex process that drives exponential growth in bacteria, research can develop strategies to optimize microbial growth in industrial settings, combat infectious diseases and harness bacterial metabolism for biotechnological application. Now, we shall learn the mathematical expression of exponential growth phase. Exponential growth phase is a fundamental concept in microbiology, describing a pattern where microbial population increase at constant relative growth rate over time. This growth phase is characterized by rapid and continuous cell division under optimal environmental condition, leading to doubling of population size at regular intervals. Understanding the mathematical expression of exponential growth in microbe is essential for studying population dynamics, predicting growth behavior and optimization various application in biotechnology, medicines and environmental science. The mathematical foundation of exponential growth phase in microbial population is based on exponential functions, which describe a quantity that increases or decreases at constant relative rate over time. The general form of exponential growth equation is n represent the number of population size or number of population size at any time. So, n t is equal to n 0 e to the power r t. So, here n t represent the population size at time t, n 0 is the population size at time 0, r is the intrinsic rate of increase of growth rate, e is the base of natural logarithm, approximately the value is 2.71, t is the elapsed time. Now, mathematical basis of exponential growth. In this equation, e to the power r t represent the exponential function, with r determining rate at which the population size n t increases over time. The greater the value of r, that is the growth rate, the faster population grow exponentially. Conversely, if r is negative, it indicates exponential decay, where the population decreases over time. To have extensive understanding of the equation related to exponential growth, a detailed exploration of component of exponential growth equation along with their interrelationship should be studied. So, let us break down each element and discuss how they contribute to understand microbial population dynamics. First is initial population size, that is n 0. So, the initial population size n 0 represent the number of microbial entity, might be number of cells, might be number of organisms present at the beginning of the growth phase, typically at time equal to 0, t is 0. This parameter sets the starting point of exponential growth and significantly influences subsequent population dynamics. The value of n 0 can vary depending on factors. The most important factor is the inoculum size, or the amount of number of cells that is present at time equal to 0, that is called the inoculum size, that is the starting point, initial condition of the growth medium and the environment where the growth occurs. Then is intrinsic growth rate. The intrinsic growth rate is a key parameter that determine the rate at which the population size increases exponentially over time. So, r is just the rate determining step. It represents the specific growth rate of microbial population. It reflects the efficiency at which the microbe convert the available resource into their biomass. The value of r is influenced by various factors, including environmental conditions. So, what are the environmental conditions? Temperature, pH, nutrient availability, as I have discussed before, and secondly genetic characteristics of microbes, and third physiological state of the population. So, these are the three main important factors that affect the growth rate. So, what is the interrelationship with number of population at time t? The population size at any time n t is directly proportional to n 0, that is the initial population, and the exponential function, that is e to the power r t. A higher r value result in stiffer growth curve, leading to faster population expansion. Conversely, a lower R value indicate a slower growth kinetics. Third important component is time or time t. So, time t in the exponential growth equation represent the elapsed time during which the microbial growth occurs. It is typically measured in hours, minutes or other time units depending upon the scale of observation. T is a function of e to the power r t. So, t is a continuous variable that influences the magnitude of e to the power r t, thereby affecting the rate at which the population size increases, that is n t. Then exponential function e to the power r t. The exponential function e to the power r t is a mathematical expression that encapsulates the exponential growth pattern observed in microbial population. It describes how the population size n t changes over time t based on intrinsic growth factor r. So, now the exponential function e to the power r t with relation to n 0 or the initial, r that is the rate and t that is the time. So, the exponential function e to the power r t is influenced by r and t with n 0 that is the initial population serving as the initial condition. It governs the exponential increase in population size, where rate of change at any time t depends exponentially on r and the cumulative effect on growth over time. Then interrelationship and prospect of individual entities in the growth equation are influenced by the initial population size. So, what are the different factors that influences the initial population size and how they affect the other factors? So, first we discuss the influence of initial population size on impact on the growth dynamics. Okay. So, n 0 that is the initial concentration sets the baseline for population growth, with larger initial population potentially reaching higher population sizes at later time under favorable growth conditions. Another impact is dynamic adjustments. Changes in the initial population n 0 can influence the onset and duration of the lag phase, where microbial population adapt to new environmental condition before entering the exponential growth. Then the second part, the influence of intrinsic growth rate r on the rate of population expansion. So, r determines the speed at which the population size increases exponentially. Higher r value indicate faster growth rate and shorter doubling time. Second criteria that influences the intrinsic growth factor is environmental sensitivity. So, here the growth rate, intrinsic growth rate is sensitive to environmental factors such as nutrient availability and temperature, which can modulate microbial metabolism and growth kinetics. The third is the influence of time. So, influence of time, here time t governs the progression of exponential growth, influencing the magnitude of population increase over successive time interval. This is called temporal dynamics. T is also critical for predicting population size at specific time points, guiding experimental and theoretical study of microbial growth dynamics. This is the most important application in predictive modeling. 4 that influences is the exponential function e to the power r t. E to the power r t encapsulates the cumulative effect of exponential growth reflecting the continuous doubling of microbial population over time. This is called as exponential amplification. The exponential function also provides a precise mathematical framework for describing and predicting microbial growth pattern, essential for modeling and simulating population dynamics in diverse environment. This is called as modeling precisions. Now, let us discuss the application of exponential growth models. Exponential growth models have diverse application over various fields. Biotechnology. In industrial biotechnology, microbial cultures are often grown under controlled condition to maximize biomass production for synthesis of biofuels, enzymes, pharmaceuticals and other bioproducts. Optimizing growth condition based on exponential growth model help enhance productivity and efficiency in the bioreactor system. Other application of exponential growth models are in the environmental science. In environmental microbiology, exponential growth models are used to study microbial population dynamics in natural environment such as soil, water and sediments. Understanding microbial growth rate and dynamics aid in accessing nutrient cycle, population degradation and ecosystem resilience. Other applications are in medical microbiology, where the exponential growth models are crucial in clinical microbiology for studying bacterial infection and antibiotic efficiency. Predicting bacterial growth rate and response to treatment help guide therapeutic strategy and manage infection diseases or infectious diseases effectively. Another application of exponential growth model is in medical ecology. In medical ecology, exponential growth models provide insight into community dynamics, species interaction and ecosystem functioning. Studying population growth kinetics help elucidate the role of microorganism in biogeocycles and ecological processes. While exponential growth models provide valuable insight into microbial population dynamics, several factors and limitations should be considered. First is environmental variability. Microbial growth rate can vary significantly in response to environmental fluctuations, nutrient gradients and stressors. Variation in R due to environmental factor can impact the accuracy of growth predictions based upon the exponential models. Second limitation is carrying capacity. Exponential growth assumes unlimited resources and identical environmental conditions, which rarely occur in natural ecosystem. The concept of carrying capacity, that is K, reflects maximum population size that a habitat can support sustainably, influencing microbial growth beyond exponential phase. Third limitation is population heterogeneity. Microbial population often exhibit heterogeneity in growth rate due to genetic diversity, phenotypic variability and spatial gradient within the ecosystem. If exponential models may oversimplify complex population dynamics and interactions among the microbial species. Let us discuss some future research directions in the mathematical model of growth, microbial growth. First is integration of omics data. Advancement in high throughput sequencing and omics technology enable the integration of genomic, transcriptomics and metabolic data with growth kinetics model. This approach enhances our understanding of microbial physiology, adaptive responses and metabolic pathways underlying the exponential growth. Second is multi-scale modeling. Developing multi-scale model that incorporate microbial interaction, spatial dynamics and environmental variability will provide more realistic prediction of microbial population dynamics in complex system. Third is dynamic environmental modeling. Incorporating dynamic environmental factors such as nutrient, pulses, temperature fluctuations into growth model can improve the accuracy of prediction and simulations in changing the environmental conditions. So, let us have some conclusive remark on mathematical modeling of microbial growth. The mathematical expression of exponential growth in microbe provide a quantitative framework for understanding population dynamics, growth kinetics and metabolic activities. Exponential growth model play a pivotal role in diverse disciplines include biotechnology, environmental science, medicines and ecology. So, the last conclusion is that the component of exponential growth equations represent fundamental parameters that conform or govern the microbial population dynamics. Their interrelationship define the trajectory of population growth from initial conditions n0 and intrinsic growth rate r to cumulative effect on the exponential growth over time e to the power RT. Understanding this component enhances our ability to model, predict and manipulate microbial growth in various applications spanning biotechnology, environmental science, medicine and ecological study. Continued research into the factors influencing initial concentration or initial rate and time will advance our understanding in microbial physiology and adaptation strategies in response to changing environmental conditions. By refining our understanding of the parameter influencing microbial growth rate, researcher can optimize microbial processes, manage microbial community and address global challenges related to health, sustainability and ecosystem resilience. Continued interdisciplinary research and technology innovations will further advance our ability to model and harness microbial growth dynamics effectively. So, the topics that will be discussed in the next session will be measurement of growth and growth health. The learner shall learn about the process crucial for understanding microbial physiology, population dynamics and various applications in biotechnology and medicines. Growth health on the other hand measures the efficiency with which the microorganisms convert nutrient into biomass and desired products. It quantifies the amount of cellular biomass or product synthesized per unit of substrate consumed. Both growth and growth health are essential parameters in studying microbial metabolism, optimizing industrial process and evaluating microbial response to the environmental changes. So, I am really excited to share this information with you and I am looking forward for further discussions. Thank you for joining the session.